All right, here we go with our screencast on nucleic acids, which of course contains the best molecule in the entire world, DNA. So nucleic acids are fourth type of macromolecule. Their function is pretty narrow. They are made for one thing and one thing only. And that's to store, transmit, and express hereditary information. So this is our DNA, our RNA, our genes. Okay, so the cool thing is that nucleic acids have a relationship with proteins in that the amino acid sequence of a protein or a polypeptide is programmed by something we call genes. And genes are made of DNA. Which is a type of nucleic acid made of a monomer called a nucleotide. And so we're going to get into that a little bit more. So there are only two types of nucleic acids. There's DNA and there's RNA. So these are kind of the least diverse of all of our macromolecules. DNA is pretty impressive. It provides directions for its own replication. And DNA also directs the synthesis of RNA, and through RNA controls the synthesis of proteins. So nucleic acids are pretty much like the master puppets of the whole thing because they control the proteins and the proteins control everything else. And then protein synthesis occurs on organelles called ribosomes. And this just kind of gives like a whole little outline of what we just talked about. So here we have DNA, it makes RNA, RNA moves out of the nucleus, and on the ribosome, we have amino acids. They are put together to make a protein. So let's talk about nucleic acids. So nucleic acids are long polymers, and they're called polynucleotides because they're made of monomers called nucleotides. Each nucleotide or the monomer consists of three parts. One, a nitrogenous base. Two, a pentose sugar, which means it has 5 carbons. And 3, a phosphate group. So if you look at a nucleotide without the phosphate group, it's called a nucleoside. So let's have a look at that. So here is a polymer of DNA, or it might be RNA. You actually can't tell. And here is my nucleotide with its 3 parts. So it has 1, a nitrogenous base. That's this part here. 2, a pentose sugar. It has 5 carbons. And then 3, a phosphate group. Okay. And the numbering here, when we talk about carbon and numbering, the numbering here is significant. So the nitrogenous base is attached to the number 1 carbon. And then the number 3 carbon and the 5 carbon are also going to play a pretty essential role in DNA and actually the formation of this polymer off to the right. So this phosphate group is attached, as you can see, to the 5 carbon. So it's called a 5' phosphate. So here's my phosphate. And because it's attached to the 5' carbon, this is the 5' end of my molecule. The next, and we're going to talk about how you make a polymer in just a minute. The next nucleotide, see here's 1 nucleotide, here's another nucleotide, here's 3, here's 4. The next nucleotide is attached to the 3 carbon. So we go 5, 3, 5, 3, 5, 3, all the way down. Okay, and we call that our sugar phosphate backbone. Let's look a little bit closer at our nitrogenous bases. So these are our bases. Okay, and again, let's look at there are 5 different types of bases. So there are 3 pyrimidines, cytosine, thymine, and uracil. And there are 2 purines, adenine and guanine. Okay, we'll talk later about how to tell them apart, but pyrimidines, you see, have one ring, and the purines have two rings. Okay, and the excuse me, the way I kind of remember them is if you cut a pie. Obviously, you don't spell pie that way, but for so cytosine, uracil, and thymine are pyrimidines. Okay, so that's kind of how I remember them. And then if you look, one other thing is that thymidine or thymine is only present in DNA, and uracil is only present in RNA. And you may be wondering, Dr. K, how do I tell the difference between DNA and RNA? And if you remember back a few slides, DNA stands for deoxyribose. Let's down here and look at the sugar. So the key is telling DNA from RNA, the key is in the sugar. So they both have 5 carbon sugars. Okay, pentose sugars. And the sugar is ribose. Again, there's that OSE. So DNA is deoxyribose, and RNA is just straight up ribose. And how do we tell the difference? You actually look here at the number 2 carbon. Ribose has a hydroxyl group on the number 2 carbon. Deoxyribose, D means without, oxy means oxygen. So deoxyribose is without an oxygen. So on the number 2 carbon, there's just a hydrogen. There is not a hydroxyl group. So that, my friends, is how you tell the difference between DNA and RNA. As we said before, the nucleoside is just the nitrogenous base in the sugar. We talked about the pyrimidines and the purines on the previous page. Okay. In DNA, the sugar is deoxyribose, so minus that oxygen. RNA is ribose, and a nucleotide is the nucleoside plus the phosphate group. Kind of review of the previous slide in words. Now, to make a polymer of a nucleotide, they are linked together, as is every other polymer, by dehydration synthesis. Okay, we'll get to that on the next slide. So nucleotides are joined by covalent bonds that form between the hydroxyl group of the 3' end of one nucleotide and the phosphate group on the 5' carbon of the next. Okay? These links create that backbone that we had a couple slides back of sugar phosphate units, and the nitrogenous bases kind of stick out in the middle. Okay? The sequence of the bases along the dRNA or the RNA polymer is unique for each gene, so the order of the bases. And that's what gives us our, well, everything, actually. So let's look at the dehydration synthesis and how we extend our nucleotide. So here I have a nucleotide. Here's that 5 carbon. Here's my 3 carbon. So here we have, excuse me, here's my other nucleotide that's going to come in. So we're going to remove the hydroxyl group from here, the hydrogen from here. We're going to remove that water molecule. Okay, so they call it a condensation reaction, but that's the same thing of dehydration synthesis. Okay, and then that allows the phosphate group to be joined to the 3' carbon here, and then we have another 3' hydroxyl at this end, and we could bring in another nucleotide here. And the bond formed, because, and then there's our water. The bond formed is called a phosphodiester bond. So remember, in carbohydrates, it was a glycosidic linkage. In proteins, it's a peptide bond. And in nucleic acids, it's a phosphodiester bond, all formed via dehydration synthesis. So A little bit on the structure of DNA and RNA molecules. RNA molecules are single, that is a typo. Oh my goodness. Polynucleotide chains. I did not write that. DNA molecules have two nucleotide chains that kind of spiral around together, forming a double helix. Okay. In DNA, the backbones are kind of run in opposite directions, 5' to 3', and we'll show that on the next slide. And this is called anti-parallel. So it has two sides, right? And the nucleotides are in the second. So one goes 5' to 3' in this direction. The other one goes 5' to 3' in that direction. Okay. And one DNA molecule is very long and it can include many, many genes. When we're making our backbone, the nitrogenous bases in the DNA pair up and hold the two sides together. There's one side, there's the other side. The sides are held together by, guess what? Hydrogen bonds. Okay. On one side of the ladder, adenine will always pair with thymine. So if there's an A on this side, there's going to be a T on the other side. There's a T on this side, there's going to be an A on the other side. And guanine always pairs with cytosine. There's a C on this side, there'll be a G on the other side. There's a G on this side, there's a C on the other side. Okay, this is called complementary base pairing. In RNA, thymine is replaced by uracil, so adenine pairs with uracil. And let's have a look. So here you can see the two sides. Here's one side, there's the other side of the backbone, and they run in opposite orientation, 5' to 3', 5' to 3'. And you always read 5' to 3'. Here's a close-up. You can see G pairs with C, G pairs with C, A pairs with T, C pairs with G, etc. Okay? What else? Oh, so just a couple of quick notes here at the end. So DNA and proteins serve as tape measures of evolution. You can kind of see how things evolved and see which organisms are related by looking at their DNA, even by looking at their proteins and their molecules. Because these sequences are passed on from parents to offspring, as organisms evolve, they continue to share DNA sequences with their ancestors or ancestral species. So two species that are closely related are way more similar in DNA than more distantly related species. So like our DNA is more similar to a chimpanzee than our DNA is to, a cucumber, okay? But you can still see similarities, okay? Molecular biology can be used to assess evolutionary kinship, not just like who's the father of the baby, but like how closely are we more closely related to chimpanzees or to apes? Are we more closely related to alligators or to crocodiles? Okay, we can tell that by looking at our DNA and our proteins. It's nuts. All right, so a little bit of review here. Again, the more organization you have, higher levels of structure, more complex structure, new properties, new functions. Organization in general is the key to the chemistry of life, or biochem. Everything is done in a very orderly fashion, and we're going to see that as we go through the rest of the year and look at different metabolic processes. Okay, the last two slides here are just kind of summaries of what we've talked about so far. And remember, all of these slides are available to you on Canvas. So you can just go there, download these slideshows. And so because this is a good thing to have a screenshot of, so we're reviewing carbohydrates and lipids. And this is a good thing to have a screenshot of. So review of proteins and nucleic acids. All right. The end. I love DNA.